In a previous webinar, we have discussed the basics of environmental monitoring plan. When we focus two aspects of the plan. First, the key elements of the environmental monitoring plan, and then how to maximize the efficiency of the environmental monitoring plan. We know that a complete environmental monitoring plan should have several components. The first component is the risk assessment and the reasoning for your environmental monitoring plan. And secondly, in the actual sampling plan, you need to define when and where, how often to sample, how many samples to take, and what to test. And also the environmental monitoring plan should have acceptance criteria that define what are acceptable and what are considered out of a specification. When things happen like pathogen positive or out of specification indicator enumeration result, what will you do? And then finally, environmental monitoring plan should be reviewed and verified in a regular basis. Today, let's take a further look at the four aspects of an environmental monitoring plan, namely Zone 1 testing, how to handle positive testing result, and what to do when pathogen positive results are recurring and persistent, and then data trending to evaluate if your environment is under control. For the discussion in this webinar, we reference FDA draft guidance in control of Listeria monocytogenes in ready-to-eat foods. The example of testing organisms are Listeria species and the Listeria monocytogenes. FDA recommends testing for Listeria species because doing so will detect both Listeria and Listeria monocytogenes. And this will help to evaluate the situation when pathogen or presence of Listeria may lead to contamination of Listeria monocytogenes. But you can apply the principles to other indicators or pathogens that you monitor. It is important to know that an EMP should be developed based on your own risk assessment. And there's no such thing called one size fits all when it comes to monitoring your own environment. We will discuss FDA guidelines on Zone 1 and Zone 2, non-food contact surface testing, and handling Zone 3 and Zone 4 testing results. You can reference that to Zone 2. Zone 1, we know, is a food contact surface. And Zone 1 testing is a sensitive topic. Many people are concerned with Zone 1 pathogen testing and the implication of a positive pathogen result. FDA does recommend Zone 1 testing for Listeria species. It is important to remember that a positive test result for the presence of Listeria species indicates the potential for contamination with Listeria monocytogenes and suggests that conditions that are suitable for the survival or growth of Listeria monocytogenes. However, a positive test result for the presence of Listeria species does not automatically establish the presence of Listeria monocytogenes. You might ask, why should we even consider testing Zone 1 for pathogens? Of course, a big reason is that it is recommended by FDA to perform routine Zone 1 testing for unlisted Listeria. There are other instances where it would be beneficial to sample Zone 1 for pathogens. For example, you have a new production line and has not been used, or you relocate existing production line, and before school production, that's a good time to test for pathogens just to get a clearance or give you suggestions to sanitize better. And when there's a construction or other major event that can potentially impact the environment, when you have pathogen positive result from food sample testing, and then sometimes when you test zone 2, 3, or 4 for pathogens, you get a positive pathogen positive result, and you are wondering if that pathogen might migrate into zone 1. And then when you have a troubling trend in your indicator testing, and finally, zone 1 pathogen testing may be warranted in joint investigation or root cause analysis. So when should zone 1 surface be tested? The result from pre-operation testing will assist in identifying situation in identify any sanitation weakness. This can be particularly useful when you sample for the contact surface before you put it into production. Zone 1 testing during operation is recommended by FDA, in particularly 3 to 4 hours into production or just prior to cleanup. This testing will give you great information on the spread of microorganisms or pathogens through your specific processing environment. A few important things to keep in mind, though: a positive sample might not be the harborage site for the pathogen, and any positive result will require follow-up actions. So it's important to plan ahead. Post-sanitation testing is important to evaluate the sanitation effectiveness. We discussed the why and when we should test in zone 1 for pathogens. Now let's get into how to carry out the sampling in zone 1. This is where many companies are worried. Many questions arise when testing food contact surface or pathogens. Following these steps can put into questions to rest and ease the mind of all involved while taking zone 1 samples. First, make a complete plan. And the plan should include corrective action procedures. If you have been if you have convinced your management to test zone 1, then you should present a well thought out game plan and all the follow-up actions in the event of pathogen result. And it's important that you involve other teams in the plan because we're going to discuss later, because all the follow-up actions is not just falling to the quality control, quality assurance people. It will impact operation, product release as well. Having a written and documented test and hold procedure is crucial when you're considering zone 1 pathogen testing. In creating these procedures, involvement from the operation team, like materials team, schedulers, and warehouse team is very important. Having all teams involved ensures the food that pass over the surface that are to be tested is placed on hold and not released until a negative result is generated. Again, it is best that you plan out a response plan before you receive zone 1 positive result, basically before you even do the testing. A checklist like this can guide you through the corrective action process. For example, when you do receive positive result, and we will go step by step how to handle it. But this is a checklist that you can look into. You want to examine the equipment, review if any design or executive or execution flaws, and review your HACCP and the food safety plan, and conduct intensified sampling and testing of the site that represent a potential source of a pathogen, and then collecting samples several times during the production to identify the source of contamination, And you also want to test the upstream from the positive food contact surface to help identify a source of contamination, and check maintenance record or for modifications or for changes, and then interview and observe sanitation. Many times this is an important part of investigation. You want to find out if any deviation was made during the sanitation process. And the review production, maintenance, and sanitation procedure to determine if there's any problems or things that happened, made the modification that prevented, that promoted insanitary conditions. And of course, identify the source of organism that caused the contamination, and review traffic patterns, equipment layout, and then whether the personnel who did the sanitation are adhering to their sanitation SOPs, and then take appropriate action. And remember to document all your actions. It is normal to feel relieved when testing result of Listeria or pathogen or other pathogens for food contact surface are negative. But if you have an action plan in place, you will feel less stressed when the results, are reported as a positive. All you need to do is to take a deep breath, and take out your action plan and get to work. What is showing on the screen now is recommended response to Listeria detection from food contact samples. But again, is from guideline. You can modify it based on your own process and your own risk assessment result. So when you receive notice that you have pathogen positive result on your food contact surface, the first step we want to do is clean and sanitize the area of where the positive samples were collected, and also surrounding area. And then retest the food contact surface and the surrounding area during the next production cycle. And at the same time, you want to trigger the comprehensive investigation of the event. If the testing result come back negative, that means all the Listeria testing negative, then you assume the problem is resolved and you can go back to production and then your routine monitoring. If any of the results come back positive, that is your second positive, then you should start with intensified cleaning and sanitizing for 3 consecutive days. And also start intensified sampling and testing for 3 consecutive days. And meanwhile, you want to hold and test your product for Listeria monocytogenes. If the result of your product testing is positive, then the product should be reprocessed, diverted, or destroyed. And then you should continue with the comprehensive investigation. If the second positive result, you test all the product and all the 3-day food contact surface testing result are negative, then you can go back to production and the routine monitoring, and then you can release any held product. However, if you get another positive, if any of the results are positive, then that's the point, that's your third positive. And that's the time you should stop production, consult expert, whether internal expert or external expert, for comprehensive investigation. You want to escalate the intensified cleaning and sanitizing, that including like steaming all the equipment, and then conduct intensified sampling and testing. You want to expand the comprehensive investigation and hold and test the product. and then continue with your comprehensive investigation. And for the product that has the positive, then again, reprocess, divert, or destroy. If after all that effort, you resume production with product hold and test in place until 3 consecutive operation days of product and for the contact surface are negative. But still, even after that, you still want to continue the investigation. The goal is find the source of the contamination. So what do they mean by intensified environmental sampling? It typically means that when a positive is found, vector swabbing is conducted using a starburst pattern, so that the 36-degree, like 3-dimensional around the site where the positive pathogen result will detected. And if additional positive find, the vector swabbing should be expanded to determine the extent of the contamination. So in that case, any item is suspect. And any open cabinet, open panels, wells, and legs of the equipment, or the things move in of the area as suspect. And you want to swab out that including the belts and drains, the cracks, drips, joints, and you definitely want to break down the equipment to find if any dead corner that can potentially harbor the pathogen. The vector swabbing is used so that we don't forget to look up and above the product and below the product, right? And looking for niches and the pathogen size. And that including any drips, condensate, or airflow over the product. You want to sample at least 3 days consecutively after a vector swabbing, because sometimes after intensive cleaning and sanitation, the pathogen source is only slightly inhibited, but it may still present. So continue sampling after the vector sampling will help you detect if that's the case. And then what do they mean by intensified follow-up sampling testing that including the product, right? If a food contact testing positive for the presence of Listeria, FDA recommends that you corrective action procedure specify when to determine if the Listeria species is Listeria monocytogenes. Then it should be risk-based. In general, the greater the risk of foodborne illness present by the food being produced on the food contact surface, then the greater the importance of determining whether the Listeria is actually Listeria monocytogenes. And Listeria monocytogenes is ranked as either serious hazard in food for the general population or severe hazard in food for the restricted populations. And the hold and test procedure must be carried out in the case when you do find the food contact surface has Listeria monocytogenes. When making product sampling plan, you can reference guidelines and determine to sample whether, take 15 samples, 30 samples, or 60 samples. As shown here, N is the number of samples to be tested. C is the number of samples that can be positive. Basically, you have to receive all negative results from the food you tested. So what do they mean? How you choose which testing plan? That depends on the condition of the food prior to consumption by human. If there is a decrease of the number of pathogens, for example, the product will be heated prior to consumption, hereby killing the pathogens, then you can use an equal 15 sampling plan. If the conditions prior to consumption are not likely to change the number of Listeria monocytogenes or other pathogens, For example, the organism will not die off, but will also not multiply, then you may consider using the n equal 30 sampling plan. If the condition prior to consumption could result in an increase in the number of Listeria monocytogenes, for example, the food will be held under a condition in which Listeria monocytogenes can grow, like ambient or refrigerated condition, then you want to consider the sampling plan to be n equals 16. When conducting an investigation, it helps to know where to look for to find the source of pathogens. I recommend you have some kind of list you're looking for. So when it's time to act, and people can go through the list and have an organized effort, try to find or sample for the pathogens. And this is a non-exhaustive list. It really serves as an example of potential sites. For example, the ingredients and the processing material might contain the pathogens, for example, raw milk, raw produce, and compressed air, any production solutions, and raw meat. And certain food contact surfaces that might harbor bacteria. For example, the fibrous or porous type of conveyor belts. I have seen some of those. And then the filling or packaging equipment in the belts, peelers, and containers, and sometimes the slicers, dicers, shredders, if not properly cleaned, or if there's any cracks, any bolts that's not cleaned properly, they can harbor the pathogens. And of course, utensils, gloves, and non-food contact surface usually have more potential to harbor pathogens, maybe because they are not as cleaned as other equipment, and also most of the time because they may be complex, right? Like in the hollow roller for runs, it's pretty hard to clean. The equipment framework, and especially if the framework is wet and rusting, open bearings within the equipment, and condensate dripping pans, which the condition may be wet and support bacterial growth, and other equipment depends on your facility. The control panels and switches, because a lot of people touch it, it can be a potential place for high bacterial count. And then in the rest of the plant environment, we know, you need to look for pathogens on the floor, especially if the floor has cracks and crevices in the walls and drains and ceilings and washing area, and anything that can hold some water is potentially subject to bacterial growth. Okay, it's when you talk about the contamination, you know where to look, and then you sample the places that are suspect. So what are the events that can lead to contamination? And try to figure that out. It's like solving a puzzle, which can be difficult. So here are some examples of scenarios that can lead to Listeria contamination. It's good to reference what people have found in the past. What usually happens is one of these change. Whenever you have a change, whether it's a packaging line change or a used equipment is brought from storage or from another plant and then installed into the process flow, and it carry the bacteria with them if they're not sanitized properly, or when you have equipment breakdown, and then sometimes the construction itself or major modifications can expose to some bacteria that was in the equipment before. And then if there's a drain backup, that can be a potential source. And then sometimes the product is called or high up in the equipment. I actually haven't seen this before. A piece of dough was stuck in the equipment. Imagine, bacteria or fungi sometimes will have a happy time to grow in there. And many times though, improper sanitation is to blame. Like, sometimes the employee is not properly trained, or if you have outside sanitizing team, they're not familiar with your operation or your control. And if there's a period of heavy production, which make it difficult to clean the floor for holding the coolers as scheduled, there is a scenario that increased production cause you to perform wet cleaning of lines that have been taken down from production in the same room as the lines that are running the product. Talk about the cross-contamination potential there. And then there's scenario that equipment parts, like screens or parts that are clean on the floor. I think this is actually happen not rarely. Sometimes there's you take the equipment part down and you don't put on a card or something, put on the floor. Guess what? The floor is the last thing. If the last thing you clean, then you subject the equipment to attract bacteria on the floor. And then of course the waste bins in the ready-to-eat area are not properly maintained. Cleaned or sanitized can cause contamination as well. And cross-contamination can happen when you're not aware. You're not aware what you're doing can cause the cross-contamination. One of the big thing is the personnel who handle the ready-to-eat food test the surfaces or equipment likely to be contaminated, like the floor, the trash can, and then they don't change glove. Or when raw or under processed food are placed in the area that designated for cooked food, if the production flow is not well controlled, this actually happen quite often. Or personnel that work both areas, work raw food area, work cooked area, this should be, things like that should be addressed in the food safety plan, and you should have production well designed. But when you're short-staffed, when you're busy, you move people around, that's when things may happen. Personnel handling ready-to-eat food can come into contact with contaminated items, right? Or and this the big thing is sometimes the indiscriminated use of high-pressure hose in cleaning, which is documented to cause problem, like you're splashing everything on everything else. Okay, so we power through the process of handling food contact surface positive result. So after that, handling of non-food contact surface pathogen positive result should be relatively easy. When you receive a notification of positive result on the routine sample for non-food contact surface, you need to pay particular attention to cleaning and sanitizing that site and at the end of production, and retest the surface and the surrounding area. For example, you conduct intensified sampling and testing and at least 3 hours into the next production run. And then if the result of this year testing all negative, then you can go back to production and the routine monitoring. If any of the results come back positive, then you should conduct intensified cleaning and sanitizing, follow the same principle, including the vector sampling, and then conduct intensified sampling and testing of the surface. If all tests coming back negative, then continue production and then go back to routine monitoring. If you have a third positive, then you should start root cause analysis. You escalate the mitigation effort and then consider consulting either internal expert or outside consultants. What happens if you have recurring positive results? And what you do with it, right? It's a troubling trend. And what actually that means? It usually means that the routine sanitation procedure are inadequate. People are not doing a good job in cleaning up. It can also mean there's a placental harborage of the organism that you detected, whether it's Listeria or whether Listeria monocytogenes or whether it's Salmonella. And in this case, you have an increased risk of contamination from zone 2 to zone 3 and eventually to food. And reoccurring of positive results also indicate a potential presence of resident strain. In those instances, the organism have persistent equipment for a while. In some cases, their records are 10 years or more. And we have the industry have been using the transient strain versus the resident strain. Basically, a microorganism that is found in passing through a routine analysis. And then after repeated sampling, the specific strain is not found. The resident strain is, as the name indicates, the microorganisms take up the residence in a manufacturing environment. These mechanisms may have formed the biofilm, which make it extremely difficult to clean. And the strain had been found repeatedly in a processing environment. Notice that the word that we use here is a strain. Strain is sub-species level. So detecting Listeria repeatedly, you don't know whether it's the same strain or not, unless you do genotyping or strain typing. And here is an example of finding a resident strain. So the situation is they are presumptive positive found on floor in a mechanic shop. The sample taken to confirmation to determine if it is specific strain. And then the plant performed vector swabbing, and they found heat on the walls adjacent to the positive floor sample. And more vector swabbing due to the second positive, right? That's the procedure. And then they found another heat. This time it's in the hand washing sink. And consistent heat in the sink after vector swabbing and follow-up swabbing on the days after the intensified situation. The crew performed the corrective action. They reviewed the sanitation document and practices, and then they disassembled the sink for intensified cleaning, and then they find the potential harborage point behind the sink where the wells were not sanitary. So the solution is update sanitation SOP, and the easier solution, just replace the sink and then make sure the equipment or the sink has the sanitary design. And sometimes the corrective action just don't work. In spite of every effort to monitoring, sampling, and correct environmental issues, including deep cleaning the equipment, deep cleaning the floor, the walls, and everywhere, the production line still tests positive erratically for pathogens or pathogen indicators, such as Listeria species. And so what do you do in that case? And such equipment, should be segregated and removed until a solution is found. If you can pinpoint the equipment, that's actually a better outcome. You can remove it, put it out of service until you find the harborage. And the facility should also, think about change the sanitation frequencies, the sanitizers to make sure it address the persistence of the indicator of pathogens. And also when the corrective action don't work, and you suspect there might be a resident strain in the facility, then you should consider doing genotyping. I know there are some concerns about genotyping, but when the time for you to do it, it's better to do it. It's sometimes knowledge can help you to take proper corrective actions. So what is a genotyping? Genotyping is about using molecular techniques to characterize the genetic information of organisms. This is a below species level characterization. So genotyping can help to assess the genetic relatedness among organisms and determine if the strains are of a common source. As I said earlier, to identify to species level is not enough. You want to see if the organisms found it in the environment, if they are actually related, closely related, that can imply a common source. So when do you want to perform genotyping? You can do it during the investigation of outbreak or big contamination event. And then when there is a recurrence of pathogens in the environmental samples, when you really need to differentiate it, whether it's transit or resident strains. And then sometimes you already identified the pathogen in both food and the environmental samples, you want to investigate where it's come from, the source of it. There are basically two techniques for genotyping or molecular typing, however you want to call it. One is the pulse field gel electrophoresis, and one is whole genome sequencing. The pulse field gel electrophoresis, PFGE, is to produce a DNA fingerprint for bacterial isolate. It has been applied into outbreak investigation to provide traceability and also compare the DNA fingerprint between isolate to determine the source or pathogens. But for the last 20, 30 years, it was used by government and companies to do the source tracking. Recently, whole genome sequencing has been used to sequence a complete genome from one pure isolate and compared with another isolate. Its applications including the outbreak, pathogen detection, source tracking, and also historical analysis for pathogens that present in the facility. Because once you get the DNA sequence, you can store it in either your private database or public database, and then you can compare your new strains with the sequences in the database to identify if two or more isolate are identical in different point of times or in different facility. Companies or laboratories can create their private genome sequence database for their client, so you don't have to post it on NCBI for the comparison. It is great to monitor the presence of pathogens in facility or between facilities, and also can be used to evaluating sanitation effectiveness. Currently, FDA, USDA, and AIH, when they conduct outbreak investigation, they can use whole genome sequencing or similar technology to screen as a quick response and find the source of outbreak strains. So here's the example of how private companies can use whole genome sequencing in source tracking. A plant that almost regular detection of Listeria in the same area of the plant. The sanitation crew and FSQA people had literally torn apart that portion of the plant looking for potential harborage. They did exhaustive cleaning, but did not break the pattern. And that's the time they sent the strain out. They collect the samples, isolate the pathogens, and have a lab perform the whole genome sequencing analysis, and that result indicating that the repeatedly occurred organisms were not related. So now they redirect their effort to search what can be a regular source of contamination. Ultimately, it was traced back to poor harvest practice of a minor ingredients vendor. What happened is the toll that they used were set on the ground to make it easier to harvest. And then the entrained soil was the source of contamination, which makes sense because Listeria is common in the natural environment in the soil. So the last topic I like to touch on today is the analysis of data for trends. Remember, the goal of environmental monitoring program is to verify the effectiveness of your control programs and find Listeria monocytogenes or other pathogen harborage site if it is present in your plant, and then ensure that corrective action has eliminated the pathogen. Assume you have diligently sampled and tested your environment, then you can make the best use of your data by conducting further analysis for trend. That can help you to continuously improve your sanitation conditions. And sometimes you can use indicator enumeration data to help to assess what is the normal, meaning the normal operation, the normal sanitation, and then from there you identify outliers. And data trending can definitely assist in investigation and the corrective actions. So here example of some data trending. There's an increase in a positive environmental samples in a particular site of areas. So you repeatedly find the positive samples in the spot or nearby area. And then another trend is you find the Listeria in the same area on multiple but not consecutive sampling occasions, right? Like if it's remember, if it's transit, you clean up, it's not supposed to come back. But if you have it reoccurring, then you need to take a further look. And then another trend is you have increase in the percentage of overall positive environmental samples in the plant. So all this trend should promote you to do complete investigation, including genotyping, and then find the pathogen harborage, and that will direct your corrective action effort. In some operations, you don't see Listeria positive result often enough to do meaningful trending. In this case, you can use enumeration test of indicator organism for trending. And this is especially useful to evaluate the effectiveness of sanitation. And then there can be 3 steps involved in this practice. First, you collect data. You sample where the system is under control, and you collect at least 20 sets of samples. So you establish what is the baseline. And then you perform testing. Some people test for APC, some people test Enterobacter AC, some people test coliforms. It depends on your specific process. And perform data analysis. It sounds fancy, but it can be really simple. You can use Excel for simple calculation, or you can use some software to do that for you. And then if you want, you can set the limit. You can set upper warning limit, and then you can also set upper control limit. What that gives you is, instead of actually looking for number, you're looking for the time when you have out of specification result and use that to trend. What is shown here is a simple data trending using Excel. The chart on the left is Arabic plate count and Easter mode count from boilerplate. In this plant, the airborne microorganism does impact their production, so they monitor the air more regularly. And at least, even you don't have that situation, you probably at least do monthly or quarterly monitoring, just see what can be in fact affect your production environment. The chart on the right is a trending of all of the specification results, and they need to find out why in certain month there are more numbers of samples turn out to be out of specification, and what happened in those months. What are the changes that lead to the changes in the trend? And nowadays, there are a lot of environmental monitoring program that use software to do the trending for them. As shown in this screen, this example, you can import all the data from the lab testing, and then you can view the result by either the Zoom or by the test type or by your environmental monitoring unit to get insight of how and the data goes and if your environment is under control. So to summarize, we talk about the four topics today. Zone 1 testing is an important component of environmental monitoring plan, and the follow-up activities should be planned out and incorporated in the EMP. So just have a testing plan, a sampling plan is not good enough. It's best that you plan out what actions you would take once you receive a positive result. And the action plan for the Listeria positive or other pathogen positive results should be risk-based and progressive, like step-by-step procedures that will help your team, at ease when they handle the positive results. For recurrence of pathogens, determining transit versus resident strain is important for finding its source and eliminate it. And sometimes is maybe change the vendor or change the vendor's practice if you identify it's only a transient strain. Because if you don't take action properly, if you don't sanitize properly, a transient strain may become a resident strain. And finally, analyzing data trend can help to understand if the system is under control and can identify outliers, and it can promote improvement effort. For example, if you use indicators, if a certain day the count is high as outliers, you might talk to your sanitation crew, like what happens, how we can do better. So thank you for your attention. I will stop here. We have 10 minutes left. If you have any questions, please ask. I will try my best to answer it. But if I don't know the answer, we'll get back to you, and you can also email lab@imtech.com with your questions or inquiries.